ICH Q1 guideline provides the information and requirements for the stability studies for drug substances and the drug products. The present video is regarding the photostability testing for the drug substances and the drug products as per the draft ICH Q1 guideline. I welcome all the viewers to this video, and after watching this video you will be able to understand regarding the photostability testing requirements, how it is conducted, and what are the regulatory requirements for this. So photostability testing is to be done for the drug substances, APIs, and the drug products. Also, it is done for the force degradation study and for the confirmatory studies for light exposure requirements. The purpose of the photostability testing is to evaluate light induced degradation risk, whether the light is affecting the drug and drug product or not, whether there is a generation of impurity or whether there is a degradation risk because of the light. So this is the photostability testing. It ensures product efficacy and safety is not compromised because of the light. As we know that different factors affecting the stability of the drug substances and drug products are temperature, humidity, and light. So out of these, photostability testing provides the guidance for effect of the light. It is required during initial submission and the life cycle changes, typically done on the one representative batch. Generally, for exhibit batches, one exhibit batch stability study for the effect of light is sufficient. So when photostability testing is required to be repeated, when there is a significant change to the process, change to the formulation, change to the container enclosure system, and change in the in-use conditions, that time photostability testing is required to be repeated. Types of the photostability studies as per ICHQ1 guideline, which is in draft, it provides the types like force degradation studies and confirmatory studies. Forced photodegradation study is a part of development phase and it is a type of stress testing to evaluate and to understand degradation pathways and to help method development. Confirmatory studies are performed if risk of photodegradation is identified. Many of the times, the confirmatory studies are to be performed on one of the exhibit batch, irrespective of whether the photodegradation is there or not. The purpose of the confirmatory study is to assess impact of light through product life cycle. It supports packaging selection and labeling. Also, it provides the requirement on whether the drug is required to be protected from light, whether the manufacturing conditions are to be performed in sodium vapor lamp or not, whether the light resistant packaging is to be selected or not. Systemic testing approach. So sequential or parallel testing will be there. Directly exposed drug substance or drug product in the primary packaging and product in secondary packaging. So how the test samples are prepared? There is a testing for the direct exposure. Drug substance, drug product are open condition and these are directly exposed to the light. And then the product in primary packaging is exposed to the light, and third one is the product packaged in secondary packaging is exposed to the light, so that you will understand how the packaging system provides you the effect of light, whether the drug is susceptible or drug is labile, photolabile or not, whether the drug is protected in the primary packaging, whether the drug is protected in the secondary packaging. For example, if you are making tablet formulation or capsule formulation, then if you are packaging it into the alu blister and then alu blister you are packaging in a carton. So the capsule in alu blister packaged in the carton will be secondary packaging and alu blister will be primary packaging, while the open exposure capsules will be the directly exposed sample. Then continue testing until the product is adequately protected from the light. Now see the forced photodegradation studies. These are performed during the development phase and it is used to or it is done to understand the degradation mechanism and to prove the method specificity. There is a reference in section 2 for forced degradation conditions in the ICHQ1 guideline. If it is combined with the confirmatory study, then guideline section 8.3 required to be followed. Confirmatory photostability studies determine the need of light resistant packaging, special manufacturing and storage conditions like use of sodium vapor lamps, use of the protective packaging, light resistant packaging and in-process packs, and label precautions like protect from light. Typically done on one batch, drug substance or drug product in the actual packaging with the appropriate controls. Sample preparation and testing design. Solids spread in a dish of around with having the thickness of around 3 mm. Tablets capsule spread in a single layer. Liquids use the inert transparent containers like quartz because you cannot keep the liquid samples open. Many of the times, because of the improper sample preparation, the product may get degraded because of the oxidation rather than the effect of light. Packaging samples expose with uniform light exposure. Then analytical evaluation. So after exposure, the the formulation and the APIs can be tested or required to be tested for the assay potency, physical and chemical degradation, physical and chemical changes, and the impurity generation or formation of the degraders. For these studies, the dark controls are used as a reference. That means these dark samples or dark control samples are not exposed or irradiated with the light, so that you can compare the exposed sample versus the dark sample and then you can know whether the light has an effect or not. So base conclusions on the photostability and other stability data is done. Radiation source and light exposure. There are three options given in the guideline. Option 1, D65 outdoor daylight, fluorescent xenon metal halide. These are the lamps. Option 2, ID65 indoor daylight, cool white and near UV exposure will be there. Option 3, ambient mild light, fluorescent LED with less than 400 nm. Choose based on the product sensitivity and the study purpose. Light exposure specifications are provided for the confirmatory study and the force degradation. So you can go through the guideline and you can get this information, and whenever you are performing the photostability studies, then you should expose the samples as per the guideline requirements. For example, confirmatory study will require illumination of not less than 1.2 million lux hours and 200 watt per meter square for the light exposure, and it must reflect real world manufacturing, storage and uses conditions. If the product is found to be photosensitive, if API is found to be photosensitive, then you need to take appropriate actions like manufacturing precautions, use of sodium vapor lamps, use of light resistant containers closures, light resistant in process packs and the hold time studies packs. Then need to use the opaque coatings, opaque capsules and protection from the exposure to the light. So these are the requirements and understanding points or key considerations as per the ICH Q1 draft guideline on photostability. Thank you for watching the video.